Okay, so this is lecture 17, and in the last class we had started really talking about organelles, and we were looking at the nucleus, and we were looking at nuclear transport. So nuclear transport, there's two types. First of all, there's facilitated diffusion, and that's based on size. But then there is, for large molecules, a selective form of active transport. And so these are unique transporters because they have both a facilitated diffusion component as well as an active transport component. The active transport component is driven by this RAM GTPase, okay, which is one of these small molecular switches. And what this switch is showing us is it's allowing the cell to differentiate between the cytosol and the nucleus. And it does this by the localization of two different enzymes. The first is, just a sec. The first is the guanine exchange factor, guanine nucleotide exchange factor. This kicks GDP out and allows GTP to be bound to RAN in the nucleus. So nuclear RAN is bound to GTP. And in the cytosol, we have RAN gap, which activates the GTPase activity and leads to GTP being hydrolyzed to GDP. So now we have a way to mark both compartments. We utilize importans, binding to nuclear localization sequences for the import, and we use exportins combined with nuclear export sequences for export out of the nucleus. Okay? A theme we're going to come back to a few times, but that we really wanted to talk to when we're looking at the ER, and today the Golgi and lysosomes, same thing, is the topology of these different organelles. And one of the important things we notice is that the inside of the ER, the Golgi, and the lysosomes is actually, the chemical composition is physically similar to that of the external environment, the extracellular space. And that turns out to be important because this compartment allows proteins to fold inside the cell in a compartment that's like the outside of the cell, which is where many of the proteins that are folded in the ER, Golgi, and so on are destined. Many of these are destined for secretion, where they'll exit the cell. We do a whole topic on that coming up soon. Can't remember if it's the next one or the one after. So the rough ER then, we have this membrane which is continuous with the nucleus, and when we're looking at the rough ER, really what we're largely thinking of, even though it's not its only purpose, we largely think of protein processing. And these are proteins that are going to end up embedded in membranes, proteins that are going to end up inside other organelles, and proteins that are going to be secreted from the cell. Because that environment, remember, inside the ER is similar to that of Golgi, lysosomes, and the external environment. And the rough ER itself, the reason it looks like it does is because those little factories, not little, actually huge, factories called ribosomes that build the proteins are associated with the translocons. So they're physically attached to the membrane. Okay? And that would be a peripheral attachment. So we have these peppering the membrane surface. In contrast, smooth ER, we think of lipid synthesis, we think of calcium storage, and we think of detoxification of many drugs or other hydrophobic toxins. And so because we're going to do a short journey through the Golgi and the ER today, let's just reiterate how proteins get into the ER. They can be built completely in the cytosol and then unfolded and brought into the ER. That's one method. What we're looking at here is co-translation translocation, meaning that translation and translocation, translocation means you're bringing something across a membrane, are happening simultaneously. And both of these types of transport post-translational or co-translationally require this this ER signal sequence, which is shown in red and is at the N-terminus of the protein. And this then initiates the translocation of the protein into the ER lumen, inside the ER, where we can get modifications and folding. And one of the major modifications that we pick up in the ER is N-linked glycosylation. This is the only place where N-linked glycosylation occurs, and it's always the same block of 14 sugars that's attached to an asparagine. Okay? And the last thing we were looking at was we introduced the idea of the Golgi, and we talked about how they were first recognized in the very late 1800s, but the confirmation of their existence wasn't made until electron microscopy was available in the 1950s. And we'll look at the importance of the Golgi today. So we're going to sort of polish up a few related organelles, so sort of ER, Golgi, and lysosomes are all involved in this endomembrane pathway. We'll talk briefly about peroxisomes and then mitochondria. Mitochondria we'll spend a bit of time on, and you actually do a lab about mitochondria. I'm not sure if it starts this week or then if it's coming up soon, but you do a lab concerning this one as well. Okay, so a couple of sort of housekeeping notes. The exam's Friday, all right, so we won't have class. Again, it's open from 8:30 to 8:30. You can write in this room because no one will be in here. Make sure you maintain the academic integrity, okay, so no conferring with other students. You can utilize your textbook, your notes, and the Brightspace page, but you can't utilize external online resources. When you start, you'll have 50 minutes. It will be the same format as the last midterm. There'll be 30 marks worth of multiple choice questions, so you should have ample opportunity to finish. I'm going to have extra office hours on Thursday from 11 till noon. So hopefully that'll catch students that are sort of straddling different class times. So if you have questions outside of that, send me an email, and I'll try to either answer by email or we'll set up another time for you to meet. So here's the bad news. We are the last day of exams. So yeah, so Monday, December 20th. That's the tentative schedule. The reason I'm putting this up is, remember it's face-to-face, so you can't make travel plans until after this. The university doesn't allow travel to get you out of exams. So this might change, but I've only ever, in all the exams I've had at UVic, I've only ever had one shift. So I would expect that this will be our timing. So it gets worse. If you're in Micro 200, the exam's the day before for that. It's the first Sunday exam ever, so we're having a lot of luck this year with exams. Anyway, and I want to make it clear that I have absolutely nothing to do with the scheduling of exams. Yes? The exam is not cumulative, however. So the exam's not cumulative, but it's face-to-face written. And the exam will be 2 hours in length, so from 2 till 4. Okay, I know it's very exciting, but let's get on with the Golgi. So the Golgi, as we said, is this kind of like a stack of flattened pancakes with a little luminal space inside them. And the way they're arranged is we can split them into kind of roughly into what's called the cis Golgi network. And this faces towards the endoplasmic reticulum. So ER here. And these vesicles are a way that we move things between the ER and the Golgi. We'll talk more about that today, but also we'll do a section of that later. At the far end, we have what's called the trans-Golgi network on the far side. And again, we can see vesicles leave to go on to either other organelles, such as the lysosome, or to actually exit the cell altogether. When we think of the Golgi, one of the main things that we think about is protein processing. So here we get modifications made to the Golgi, and it's also a sorting center. So it's important for the trafficking of proteins. It basically directs them to different places in the cell. If we look inside here, the way this diagram is set up is the ER is shown on the top and the Golgi in the bottom. I'm not going to ask you to memorize the different processes carried out in the different compartments, but what happens is that if we were looking at kind of a complete pathway, proteins might move perhaps all the way through, and as they move through the different compartments, you can see in your notes there's a number of different types of modifications that occur. That big N-linked clump of sugars can be modified here, but N-linked glycosylation can't occur here. So the N-link has to be started in the ER. There's some modification in the ER, but there's further modification can occur in the Golgi. In the Golgi, we can carry out what's called O-linked glycosylation, which is through the hydroxyl group on the side chains of serine and threonine residues. And if we look here, we can see that many of these steps involve modifications of glycosyl groups or addition of glycosyl groups. But there are other protein modifications occur here as well. And so this is an overview then of the endomembrane or endolysosomal system. And we're going to spend a lot more time on this coming up. But what I want you to note here is that to the left, we see the endoplasmic reticulum. Then you can see we have the Golgi. And then we're moving through, and the next organelle we're going to talk about is the lysosome. So we can see that the other important thing to see is that things that move between these move via vesicle, not transport. So they're not exported to the cytosol and then transported into the next organelle. They move about here. And what do we notice about the interior of this? They're all the same color, which means the chemical environment is very similar. This is really important because when you fold a protein, how it folds up is actually dependent on the surrounding environment. And so we initially start the folding here in the ER. It may even complete in the ER. And we maintain that environment no matter where these proteins are going, even if these proteins are destined for secretion, as we see it up here. And so they can move through this system of vesicles between the different organelles. Yes?
Sorry, just a quick question. The O linkage in the Golgi?
Yes?
Between the hydroxyl group and the serine and threonine?
So serine and threonine are the hydroxyl groups that the sugar is added to. Yes.
Okay, thank you.
Okay, so now we're going to talk about lysosomes and peroxisomes. I've gone through and added the ebook sections to all of the notes, and those are posted separately in Brightspace. So I, for whatever reason, didn't clue into the fact that the textbook and the ebook wouldn't align. So people were asking about that, so now you can align that with the ebook. And so those are posted on the Brightspace in with the lecture notes. So there's topic 2, and then topic 2 reading list that it says with ebook, topic 3 reading list, and I'll continue to do that for the remainder of the course. So lysosomes are organelles that are important for degradation of macromolecules or big molecules. The proteins that are what we call resident proteins, those are the proteins that remain in the lysosome and are important for the lysosome. Those have traveled from the ER to the Golgi, and then they've been moved via vesicles to the lysosomes. The lysosomes are acidic, and we talked about V-type ATPases. Those are an important component of this. So the compartment itself, you can see the pH is about 5. If we consider physiological pH at 7, we know the pH scale is a log 10. That means that this is 100 times more acidic than the cytosol. So inside this, there are a series of what are called acid hydrolases. What does a hydrolase do again? Do we remember? Yeah, they break bonds by adding water to them. And what these can do is these ones can break apart, nucleases break apart nucleic acids, proteases break apart proteins, glycosidases break apart different carbohydrates, lipases break up lipids, phosphatases, sulfatases, these take off post-translational modifications, and phospholipases break down phospholipids. So what can these things break down? Everything. So they're basically capable of breaking down pretty much everything in the cell. The acidity is maintained with our V-type ATPase, and why do you think it's important that the compartment is acidic and that all of these hydrolases are what we call acid hydrolases? What do you think? Yeah.
Does the acidic environment make it easier to break apart macromolecules?
So would the acidic environment make it easier to break apart macromolecules? It might assist a bit with some of the unfolding or things like that. There's another maybe more subtle thing that's happening here to do with the hydrolases themselves. Yeah. Yeah, so the shape of the protein depends on the environment. So you're getting, I think, to the point there where these acid hydrolases function at acidic pH. And they're folded and they're structurally functional at acidic pH. At neutral pH, they don't function. So this means if our lysosome ruptures, which can happen, and the acid hydrolases are released, they don't just chew up the entire cell. So that's an important sort of safety feature. So this is a little bit strange. So they're what we call heterogeneous, meaning that lysosomes are not all the same. And the reason for that, I'm going to go all the way back to this slide. If you look at the way that a lysosome forms, it starts out as a vesicle, and there's different pathways it can achieve, and that matures, and the vesicles fuse together, and the vesicles then fuse with existing lysosomes. And so what we're looking at is this whole series of events where things are moving. So we have a vesicle, and another vesicle fuse, and then that fuses to a lysosome, which then is bigger and not quite as acidic, and then it gets acidified. And so that's what we mean when we say that they're heterogeneous. Because of the way they form, it's not just one fixed organelle, okay, as you may see with something like a mitochondria, where its size and composition is relatively stable throughout, a X number of minutes. These are constantly changing. We can think of them sort of as like soap bubbles in a sink or something, where the bubbles kind of fuse together and form bigger bubbles. That's what's happening here, except we have other bubbles kind of breaking off. And again, we'll spend more time describing that in a later section of the course, but it's something really interesting about all of the components, the ER, the Golgi, and the lysosome are constantly, vesicles are constantly being pulled out, so parts of their membrane is constantly being pulled out, and new membrane is coming in and fusing with it. So they're in constant fluctuation. All right? 3 pathways that deliver material to the lysosomes. So the first 2 are phagocytosis and endocytosis. These are 2 different ways in which we bring things into the cell. So what's happening here is the cell is pushing in and then will eventually seal off and form either a phagosome or phagocytic vacuole or an endosome. And these then fuse together and eventually fuse with the lysosome. We can also have the process called autophagy, which is shown to the right. This is a way in which the cell actually breaks down its own components. So what will happen is with time, say an organelle is no longer functional, it's not functioning optimally, you can break it down to its component parts in the lysosomes for recycling and build new mitochondria or whatever it might be, for example. So peroxisomes are not part of that ER-Golgi lysosome system. These are distinct, and these are sites where we get oxidation occurring. And oxidation can be important for a number of reasons, all right, but it's often involved in the breakdown of molecules. In this particular fluorescent image, okay, what you have is the nucleus is stained in blue, the mitochondria are stained in red, and the peroxisomes are stained in green. So we can see there's lots of these peppered throughout the cell. So oxidation occurs here utilizing what we call molecular oxygen and hydrogen peroxide. I'll show this in the very next slide. It's also one of the sites where we get oxidation of fats. And that's important for freeing up the energy from fats. So we get what's called beta oxidation in both the peroxisomes and the mitochondria. So what we're looking at in this example is here is whatever this molecule is. And this is in a reduced form, meaning it has electrons to give up. And then what happens is we utilize oxygen to oxidize this, so R has become oxidized. And we've made hydrogen peroxide. In the next step, that hydrogen peroxide can oxidize another group. So R prime is now oxidized, and we produce water as well. So we're not going to go into a lot of redox details, but we'll talk about it a little bit. Remember, when something loses electrons, it's oxidation. When something gains electrons, it's reduction. And what happens is the oxygens move from one species to another. So in the top, the electrons, which remember carry a single negative charge, they move from the RH2 to the oxygen. And in the second one, the electrons move from RH2 to the hydrogen peroxide. And so we have this 2-step process that's an oxidation for whatever the material here would be. If this malfunctions, it can lead to what's called Zellweger syndrome, okay, which leads to developmental abnormalities in the brain, liver, kidneys, and other organs, and usually results in mortality at less than 6 months. So peroxisomes are an incredibly important organelle. Okay, we're going to move on to the mitochondria. Is there any questions about any of the previous organelles? Yes?
Could you please explain what an endosome, a phagosome, and autophagosome are?
Yes. So a phagosome is formed by phagocytosis, an endosome by endocytosis, and those are the two different ways we can bring things in. So we'll look at that in a lot of detail coming up. Okay, so the mitochondria is kind of a biggie. I've cut quite a bit of the material out of this this year to give us a little bit of a break. If you're taking micro, we cover redox in a lot more detail in that course, but we will pick that up and cover it in a lot of detail in Biochem 299. So I took some of it out of here. You'll still have enough information about it that you won't have a problem getting through the labs. So, because I know we have to do a lab on this one. All right, we talked about this a little bit when we looked at the endosymbiont hypothesis, but when we're looking at the mitochondria, it's pretty cool because it has its own little genome preserved from when it was presumably a free-swimming bacterium. And most of the genetic material has been lost, but in humans, we still encode within our mitochondria about 15 proteins and a couple of other gene products. So inside the mitochondria, DNA replication, transcription to RNA, and translation all occur. Most of the proteins in mitochondria, however, are now encoded on the nuclear chromosome. They are transcribed in the nucleus, translated in the cytosol, and then imported into the mitochondria. It's important to note that the mitochondria has two lipid bilayers surrounding it, and that gives an additional sort of inner membrane compartment. The replication of mitochondria is not necessarily linked to the cell cycle, and the cell cycle, remember, is how we regulate when a cell replicates. So mitochondria are separately maintained in their numbers. In humans, mitochondrial DNA is almost exclusively maternally inherited. So if you're watching like a forensics show or something, often you'll see that they're sequencing mitochondrial DNA. That's because it's almost exclusively maternal. The DNA that's in there, that little genome. And so, kind of neat. These are where we make almost all of our ATP. As a result, there's lots of mitochondrial disorders. So if your mitochondria are not fully functional, it can lead to a whole host of disorders, depending on what tissues are lacking the energy production. And so this is just a partial list of it, but you can look them up and see there's a lot of diseases that have a mitochondrial component. So they're the little energy factories in the cell. We'll look at some of these, well, we'll look at these processes in a sort of high-level way in a few minutes, but what we have, for example, is sugars, such as glucose. The first steps, the breakdown is in the cytosol, but then they're brought into the mitochondria where the breakdown of them is completed. Over here we see our triglycerides, remember our lipid fat storage. They are broken down and utilized as energy in the mitochondria. Down here, we can see our ATP synthase pumping out a whole bunch of ATP. And so lots of ATP, not all ATP, but almost all the ATP we make is made in the mitochondria. And so they're extremely important for this. Here's like a really high-level overview just showing sort of two breakdown pathways for sugars and polysaccharides on the top and fats or fatty acids on the bottom. So there are then these different compartments, and there's an outer membrane, and then there's an inner membrane, and the inner membrane is highly invaginated for reasons we'll talk about in a minute. So where do you think that outer membrane originated according to the endosymbiont hypothesis? What do you think? Yeah.
The cell membrane?
Yeah, so this was the cell membrane or the plasma membrane of the cell, which means that inner mitochondrial membrane, oh, we can use this one here. Where did that come from? That was... Yeah, that was the bacterial PM. There is this then intermembrane space. Okay. And there is also in the interior, and so we call that the intermembrane space. And then on the inside, we call this the matrix. And so this chart is telling us the different processes that occur in each of the compartments, and it turns out that these compartments of an organelle, which is in itself a compartment, are important for the function of the mitochondria. And so we'll look at that. You can always refer back. This is quite a nice diagram showing what happens in the different compartments. So most of what we're going to be looking at now, well, actually it occurs in all three compartments. So again, I'm going to talk a little bit about glycolysis. We're not going to do this in as much detail as micro or what you'll see in Biochem 299. I just want to give you sort of the take-home or the important points of it. What you should know about these processes, these pathways we talk about, is what goes into them and what do we get out of them. And so here, this one is demonstrated or illustrated at the bottom of the, that's shown down here, is what goes in and what comes out. So this is a nice diagram. This is sort of the level I'm looking at for this. And what we can see is we start with glucose, which is a 6-carbon molecule. And 2 ATP are added to that. And then it's oxidized. And so what happens in the oxidation step described here is electrons, which we just draw like that, move from the carbon source to carriers. Electrons always have to go somewhere, and where they go is they go into these carriers, which are called NAD+. So 2 NAD+, each pick up 2 electrons, and we form 2 NADH. So again, the analogy here is with dump trucks. So NAD+ is like an empty dump truck that we can move electrons into, and then those electrons can be carried to where they're needed in the cell. When it's full, it's called NADH. So those are our 2 states of our dump truck. NAD+, no electrons, NADH, which is carrying 2 electrons. And at the end of the process, we actually have two, well, the middle and the end here, we have two steps. Just change the color. Where we're producing 2 ATP. So 2 ATP went in, 4 came out, so we have a net gain of 2 ATP. So actually, breaking down glucose in the cytosol doesn't produce much energy. We filled up 2 of our electron dump trucks, and now our 6-carbon glucose is now 2 3-carbon molecules called pyruvate. Pyruvate then is transported from the cytosol into the matrix of the mitochondria. And what we see here is 2 steps. The top is a feeder system into what we call the citric acid cycle. And the citric acid cycle, also known as the tricarboxylic acid cycle, or TCA cycle, or the Krebs cycle, all interchangeable. What happens here is we feed in a 2-carbon molecule. So our 3-carbon molecule is converted to a 2-carbon molecule, and we release 1 carbon dioxide. Carbon dioxide is the fully oxidized form of carbon, so we've taken all the electrons out of it. And once you've sucked the electrons out of the carbon, it's a waste product. We can't get any more energy out of it. And the way we're getting energy, if maybe we've missed it here, is the way we're actually going to end up getting the energy out of glucose or whatever our fuel is we largely get it by pulling the electrons out of it. And as it becomes less and less laden with electrons, it becomes more and more oxidized, and it's at the end of the process, it's carbon dioxide, which has no more electrons to take, so what do? We just breathe it out. It's junk to us. So now, we've gone from 3 carbons to 2. This goes into the TCA cycle, and all the rest of the electrons are stripped out, producing 2 more carbon dioxides. And along the way, filling up a bunch of those dump trucks, NAD+. Okay. And also a second type of electron carrier called FAD. And so here it is in detail. You don't need to know this level of detail, but it's super interesting, and you might want to look through that. Again, I think many of you have hopefully seen the TCA cycle. Well, you have seen it before, but you probably haven't memorized it. And then lastly, so this is what we end up getting when we start with a glucose and we take it through to the end of the TCA cycle. So we have a problem now in that for this process to occur, change the color. So for this process to keep occurring, I need my empty dump trucks for electrons. For this to occur, I need my empty dump truck for electrons. But what we can see is at the end, what we actually have is a whole bunch of full dump trucks. And so what happens on a building site if all the dump trucks are full? The building stops. So in this case, the building is a breaking down of sugar, so everything's going to grind to a halt. And none of these processes will continue until we empty the dump trucks. And so when we empty the dump trucks, these processes can continue. And so the way that's done is we empty them into what's called an electron transport chain. And we can imagine, with this amazing dump truck analogy that I'm obsessed with today, is that our dump truck is going to drop those electrons off at the top of a hill, and they're like big rocks, and they're going to roll down the hill, those electrons, and as they roll, they're going to be used to turn turbines. Okay? They're going to be used to do work. So they're going to fall through kind of like a water wheel. Okay, can we imagine this? We're an oldie-timey town. We see the watermelon. We're like, oh yeah. So that's what's happening. And what those little windmills are doing is they're pumping electrons across into the intermembrane space. And they're pumping them from the matrix into the intermembrane space. All right? So far we're pretty good with this analogy. Our dump truck's dumping our electrons. It's rolling through all of these pumps, powering them. And those pumps are moving protons. What kind of transport is this called? Yeah, it's active transport. We're pushing protons up a concentration gradient. So this is just a fancy transporter. And then eventually those electrons have to go somewhere to the final product, and the final product is oxygen. This is why we need oxygen. And those electrons go onto oxygen, and what happens when you add electrons to oxygen? You make, it's building up in everyone's masks and glasses as we speak, water. So we dump those electrons, they go onto oxygen, they produce water, and that's our second waste product. So we've taken glucose, we basically burn it, we use it up, we oxidize it to carbon dioxide and water. Okay? And so we can see a little bit, sort of a diagram for that on the right. Okay? And what this is showing is you can convert energy into different forms. What we're doing in this example is we're utilizing that energy from the dump truck which originated from oxidation. Remember, that dump truck was filled by oxidizing our carbon, right? Pulling all the electrons out. And we used it to do work. But you can do the same process if you take like a piece of wood and you light it on fire, where you're oxidizing that wood to carbon dioxide and water. But in that case, you're converting the energy not into work, but you're converting it into heat and light, as we see on the left here. So that's a very similar process in that what goes in and what the end product is are very similar, but we've done different things with that potential energy. Oh, someone's phoning in to fact check me. It all holds up, I swear, look. So this is just describing the energetics of it. We're not covering that in this term because we'll do it in 299 and I don't want to spoil the surprise. But basically, each step, as it moves through that electron transport chain, each step is an energetically favorable flow of electrons. And we know if we're doing something favorable, we can link it to something unfavorable. So here the favorable flow of electrons is mixed with the unfavorable movement of protons up a gradient. And so here we can see how that occurs. And so if we have NADH, and remember we made a whole bunch of these NADH, these full dump trucks, we move 4, 8, 10 protons across the membrane. So for every NADH, we move 10 protons across. FADH2, which isn't shown on here, it feeds in here. And so for every FADH2, we move 6 protons. And that's shown here, feeding in here. So both of them join up at this coenzyme Q stage. So FADH2 can only move 6. So we're going to pick it up with what happens to this, because what we're going to do now is those protons want to go back into the matrix. That's favorable for them to move down a concentration gradient. That's favorable. We're going to link that to the unfavorable process of creating ATP from inorganic phosphate and ADP. Okay, so we'll see everybody tomorrow.